Welcome to this session. Today we are going to be discussing a key topic in molecular biology, transcription under the subject of microbiology. In this session, we will understand this topic step by step. Let's begin with the basic concept of DNA transcription. So, what is transcription? It is essentially the first step of gene expression. In this process, the information stored in a DNA sequence is copied into a complementary RNA molecule. Think of DNA as the master blueprint, double-stranded and safe in the nucleus. Transcription uses this blueprint to create a single-stranded RNA copy, which can then go out and guide protein synthesis. The entire process is carried out by a very important enzyme called RNA polymerase. Now, to understand how RNA polymerase works, we need to look at its structure, especially in prokaryotic organisms like bacteria. The RNA polymerase is not a single protein, it's a complex made of several subunits. Through experiments like SDS-PAGE, scientists identified its core components. The main body consists of two very large subunits named beta and beta prime. Then we have the smaller subunits, sigma and alpha. There's also a very small subunit called omega, which is sometimes harder to detect but plays a role in assembly. Continuing with the structure, each subunit has a specific gene coding for it and a specific function. The beta prime subunit is the largest, encoded by the RPO-C gene. The beta subunit is the next largest from the RPO-B gene. Then we have two copies of the alpha subunits. Together, these four subunits, two alphas, one beta and one beta prime, form what we call the core enzyme. This core enzyme can do the job of making RNA, but it's not very specific or efficient on its own. This is where the sigma factor comes in. Sigma is a specificity factor. When the core enzyme binds with the sigma factor, it becomes a complete machine called the holoenzyme. The sigma factor's main job is to guide this holoenzyme to the correct starting point on the DNA, which is a region called the promoter. Without sigma, the core enzyme might start transcription anywhere, but with sigma, it initiates efficiently and specifically at the beginning of genes. The most common sigma factor in E. coli is known as sigma 70. So, what exactly are these promoters? Promoters are specific DNA sequences that act as binding sites for the RNA polymerase. They are located upstream, meaning before the actual coding region of a gene. You can think of a promoter as a start here sign for the transcription machinery. In the lab, when transcription begins at a promoter, it mimics what happens in a living cell. The sigma factor we just discussed is crucial because it helps the polymerase bind tightly to these promoter sequences. The process of the RNA polymerase finding and binding to a promoter happens in distinct steps. First, the holoenzyme loosely scans the DNA, binding and rebinding. This is the promoter search. Once it finds a promoter sequence, it forms an initial loose association called the closed promoter complex. Finally, the DNA strands at that site locally unwind or melt, and the polymerase binds tightly, forming the open promoter complex. This open complex is now ready to start synthesizing RNA. Now, let's look at the structure of a typical prokaryotic promoter. They have two key conserved sequences. About 35 base pairs upstream from the start site, we have the -35 box with a consensus sequence often like TTGACA. About 10 base pairs upstream, we have the -10 box, also known as the Pribnow box, with a consensus sequence of TATA. These boxes are separated by an optimal distance of about 17 base pairs. The -10 box is particularly important for the initial melting of DNA, while the -35 box helps control how often transcription is initiated. In eukaryotes, promoters are more complex and diverse. They often contain a core promoter element like the TATA box located around -25 to -30. Upstream of that, there can be other elements like the GC box or the CA80 box, which are proximal control elements. These sequences are binding sites for various transcription factors that help assemble the transcription machinery. The start of transcription itself is denoted by an initiator element or INR. So, eukaryotic transcription requires the coordination of many more proteins at the promoter. Beyond the promoter, eukaryotic genes are also regulated by elements called enhancers. These are DNA sequences that can be located very far away from the gene, sometimes thousands of base pairs upstream or downstream. Enhancers bind specific proteins, and this complex loops the DNA to bring the enhancer physically close to the promoter. This interaction dramatically increases the rate of transcription initiation, turning gene expression up like a volume knob. The overall process of transcription can be broken down into three major steps: initiation, which we have been discussing, elongation, where the RNA chain grows, and finally termination, where synthesis stops. Let's delve a bit deeper into initiation. After forming the open promoter complex, the RNA polymerase begins to synthesize a short RNA strand about 9 or 10 nucleotides long. This initial phase is a bit unstable and the enzyme might release short abortive transcripts. Once it successfully synthesizes past this point, it clears the promoter, the sigma factor often dissociates, and the core enzyme transitions firmly into the elongation phase. Now, it's committed to reading the template strand and building the RNA. This brings us to the elongation phase. Here, the core RNA polymerase moves along the DNA template strand. As it moves, it unwinds the DNA helix ahead of it and rewinds it behind it. The growing RNA strand forms a temporary hybrid with the DNA template. Nucleotides are added one by one to the 3 end of the RNA chain, matching the complementary base on the DNA template. The enzyme continues this process steadily down the gene. During elongation, the core polymerase we talked about earlier is the key player. The beta and beta prime subunits form the active site where the phosphodiester bonds between nucleotides are formed. They are also involved in binding the DNA template. The alpha subunits have roles in assembling the core enzyme and interacting with other regulatory factors. So, each part of this molecular machine has a defined job in extending the RNA transcript. Finally, the process must end. This is the termination phase. RNA synthesis continues until the polymerase encounters a specific termination signal in the DNA sequence. This signal instructs the polymerase to stop, release the newly made RNA molecule, and dissociate from the DNA template. In eukaryotes, termination is often linked to the processing of the RNA tail. A common signal in the newly synthesized RNA is the sequence AUAAA. When the polymerase transcribes past this point, the transcript is cleaved and released. The DNA double helix then reforms behind the polymerase. In bacteria or prokaryotes, there are two well-known mechanisms for termination. The first is Rho-independent termination, which relies on a specific sequence in the RNA that forms a stable hairpin loop, followed by a run of uracils, causing the polymerase to stall and release the RNA. The second is Rho-dependent termination, which requires a helper protein called the Rho factor. Rho binds to a specific site on the newly made RNA transcript and uses energy from ATP to move along the RNA towards the polymerase. When it catches up, it unwinds the RNA-BNA hybrid within the polymerase, forcing it to stop and release both the RNA and the DNA template.
Looking at this diagram for Rho-independent termination, you can see the key features. The RNA forms a GC-rich hairpin loop structure which destabilizes the RNA-DNA hybrid in the polymerase. This is immediately followed by a stretch of adenine bases on the DNA template, which results in a string of uracils in the RNA. This weak UA hybrid, combined with the hairpin, causes the RNA to be released. For Rho-dependent termination, the Rho protein is the key. It binds to a specific site on the newly made RNA transcript and uses energy from ATP to move along the RNA towards the polymerase. When it catches up, it unwinds the RNA-DNA hybrid within the polymerase, forcing it to stop and release both the RNA and the DNA template. This covers our overview of the transcription process. Thank you for your attention.